Are you studying for the testimony?
Yes, I am.
Do you understand like the whole like SLiCE thing?
Well, I understand that it's a type of DNA engineering method that they use.
Wait, you guys are going over SLiCE?
Yeah, we're just about to.
Okay.
Well, before I start explaining, can you just read this section first?
But
And then we'll,
I mean, I feel like I'm.
trust me, this has everything.
Alright.
But after that, we can discuss it together. But just so you know, it's seamless ligation cloning. Good morning and welcome back to the Food Scientist. I'm Professor A.
And I'm his lovely assistant, Choo.
And on today's show, we will be showing you a new homemade way of cloning known as SLiCE, Seamless Ligation Cloning Extract Method. Let's get started.
Yeah. Before we do that, we have to let everybody know where we got this recipe from. Today's day is viewer's choice on our segment, and we got our recipe from Daddy Nate 08.
Thank you, Daddy Nate 08. Let's get started.
Add him on Instagram.
First step of the SLiCE protocol is to prepare your SLiCE from an E. coli recombinase A deficient strain. You will need buffer solution containing 3% weight per volume tritin X100. This is a lysine solution. Next, you will need 1 milliliters of E. coli, which were originally grown in liquid broth medium at 37 degrees Celsius, but then transferred to 2X YT media, which is more nutrient rich and able to support growth of both bacteria and phage. Next, you will need a 37-degree incubator with shaker to support maximal growth, as Juju is demonstrating. The next step is to centrifuge at 5,000 Gs for 10 minutes at 4 degrees Celsius. Now that the centrifugation process is complete, we can resuspend the cells in tritin X buffer containing 50 millimolars tris HCl solution. This is to complete the lysing procedure and to produce the cell lysates from which SLiCE is extracted from. As you can see, here is the wash with the sterilized water. And now we have our solution ready for a BCA assay method to determine the total protein concentration of SLiCE that we have just extracted.
You will then need to prep your insert DNA fragments and linearized vector DNA. We're going to be doing this by performing PCR. It's going to be a process of denaturation, annealing, and extension. So, what you're going to need is our vector. There you go. You're going to need your insert.
Now the vector is what goes into this E. coli cells, correct?
You're the professor, you should know.
That is correct.
Oh, okay. Then, you're going to need your DNA polymerase, primers, so that you can attach your insert to your vector. There you go. 3 primers to be exact.
Am I mixing right now?
I mean, knock yourself out. You want to make sure to not get any of your DNA polymerase shells in your mix because that won't be good and someone could sue you.
So essentially now what we have is the products or the reactants needed for annealing and extension during the PCR process.
Also, you want to add that's DPN1.
DPN1 is a restriction enzyme that prevents cross-contamination of methylated DNA, just to keep our DNA safe throughout the whole process as we're baking.
And then you're also going to need your DNA purification kit. after the agarose gel electrophoresis has been run.
And that is to isolate the insert and vector that we are amplifying during PCR.
You need to put some elbow grease in that.
Let me try.
Put your back into it. There you go. Ooh, 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 ooh. I thought you were supposed to be the professor. You're supposed to know how to do this.
I teach you cook.
Oh, okay.
Now we are ready for the SLiCE reaction. For each SLiCE test tube, we will need 10 nanograms of linearized vector, which is PCR amplified, an appropriate amount of insert, 1 microliter of SLiCE buffer, 1 microliter of SLiCE extract, and enough sterilized distilled water to bring the reaction up to 10 microliters. So Juju, what's your technique for prepping these cupcakes?
It's all in the wrist. You want to keep it nice and light, okay? So then you want to take your spoon. You want to take your vector and just put it over and just fill it.
Oh, I see.
Yeah. You don't want to fill it all the way because they're going to rise. So you're going to just do that, okay? And we're going to go to the next one. It's okay if you make a little bit of a mess. It's just going to burn a little bit, but you can just like peel that off. And then the next one. Okay. You know, it's funny because growing up, me and my mother made clones all the time. So this is just hitting home for me. You know, I'm getting a little emotional thinking about it. But, you know, it's great, you know, to do this. Thank you. Yes. So now we're gonna take our insert and insert it seamlessly because that's what we're doing.
Seamless ligation cloning extract method.
Our ooey gooey center, yeah. So you're gonna take it and you're gonna just place it right in the middle and try to dip it low, bring it up slow, roll it all around, poke it out, and make that back roll. Okay? There we go. We're gonna go through and do that for all of them. Here we go. Did you clone with your mother when you were younger? Was that something that you guys did?
No, I just selected for the ones that had competent vector inserted, and I would eat them.
Oh,
Yes.
well, you're the professor. I don't really know what that means, but I guess everybody's childhood is different. But yes, okay, we got 2 more. Here we go. And the last one. There we go. All right.
So now we're ready for incubation at 37 degrees Celsius of the baking tray for 15 minutes. We'll talk to you right after then.
This is a public service announcement. Breaking news. Research has found that seamless ligation cloning method is better than traditional cloning.
SLiCE uses easily accessible cell lysates to assemble DNA fragments into a vector. It's time-saving, and the reduced use of restriction enzymes alleviates the problem of the limited availability of restriction endonuclease sites. Also, you are able to incorporate 2 unpurified DNA fragments into a vector simultaneously.
Homologous recombination is a form of genetic recombination that exchanges nucleotide sequences between similar or identical molecules of DNA. Common applications of homologous recombination include double-stranded breaks, production of new sequences during meiosis, which lead to genetic variation. Homologous recombination is conserved among all domains of life, including bacteria and viruses.
Overlay regions between strands of nucleic acids form holiday junctions that lead to the crossover of nucleotide sequences and the incorporation of those new sequences into the opposite strand. Ideally, you would like to use an E. coli strain that encodes a prophase recombination system, such as the PPY strain, which includes a lambda prophase red/ET recombination system. That uses short-end homology regions between insert and vector DNA fragments. However, you can alleviate this problem by using an E. coli lab strain that is harvested at the late log phase and extract the lysates carefully on ice without the requirement of exogenously expressing a lambda prophage. The results show that SLiCE was effective in producing colonies that contained the new DNA and vector. The lab strain E. coli showed promising results when up against the commercially available SLiCE kit containing the lambda prophase recombinase system.
And now we return to our regularly scheduled programming.
Welcome back. Over the break, we incubated the cells containing the vector and insert together on liquid broth agar plates for 37 degrees Celsius. And now we're selecting for the cells that have survived an ampicillin treatment, which was contained in that liquid broth. The cupcakes that have the frosting on top of them survive the ampicillin treatment, and therefore we know that they have the correct vector inside of them. So as you can see, Jude just putting the frosting on.
Again, all in the flick of the wrist.
And now we have completed SLiCE, or seamless ligation cloning extract. We want to thank you for tuning in today.
Thank you. We're going to taste these first.
Right. We'll see you next time. on
Science is great.
The food. So that's the seamless ligation of the insert into the vector. Does that make sense?
Yeah, I think.
Alright, great. Let's go fast.
See you later, Hank.
Bye, mate.